I'm interested in the health of marine invertebrates, specifically echinoderms, so sea urchins and sea stars and sea cucumbers. And the reason I'm interested in them is because the place where I do a lot of my research is right across from the port of Los Angeles, which is super cool. But inevitably, I'm sure there's definitely like pollution and toxins associated with that. And it happens to be also the same place where a lot of these critters live. So I'm interested in understanding how that might impact their health. The way that I'm trying to look at that is by examining their microbial communities like within their bodies and whether or not they're impacted by the toxins and pollution, and whether or not that then in turn impacts the host's body, so the body of the sea star or the urchin. They are just microscopic little animals or bacteria or viruses. So we talk a lot about the human gut microbiome. So when your doctor tells you to eat yogurt to restore your internal fauna, they're talking about those really tiny little microscopic friends that are part of your body and part of your health in a lot of ways. So that's what I'm interested in looking at, but instead of in humans, it's in sea stars and sea urchins. So my understanding is that most microbes, so bacteria, viruses, tiny little animals, single-celled organisms, are harmless, but it's that like very small percentage of things that are pathogenic that will mess with you. With that though, our bodies are constantly associating with those tiny little organisms. So sea stars and sea urchins, just like us, have their own little community of microbes that they associate with, which in most cases is useful or doesn't do anything to you at all. But if you are exposed to some environmental variant, like a pollutant or a toxin, that might change the microbes in your body, and then that might set off some sort of cascade of metabolic or physiological processes that then changes your immune system or your health in other ways. My senior year of undergrad, I was studying little sea stars, and I was studying them right at the point where they turn from their larval stage into their what looks like a sea star stage. So they're really tiny, and we were gonna run this whole other set of experiments, but then they all started disappearing, and it turns out that they were eating each other. So they're like 4 days old, they're about the size of a grain of sand, and they're just going to town on their siblings. So that's what my undergrad research turned into. And then I was interested in studying that in graduate school, to sort of actually look at what their microbiome might look like when they're cannibalizing versus when they're not, or what kind of nutritional value do they get from eating their siblings versus what we thought they like to eat. However, it's really difficult to rear sea star larvae all the way through to metamorphosis. And it didn't work when we tried it last summer. So we're switching gears and we're focusing on the larval stage. Let me know when they do. They don't! At least there's a lot of troubleshooting and there's a lot of tweaking. It's frustrating, but science is bottomless. So whatever direction you can't take it in, you just tilt a little bit to the side and you've got a whole nother like ground to bury into of knowledge that you can find. When something works, it's really exciting. When you get data, it's really exciting. And honestly, just working with like live little animals that I get to see sort of like progress through their life stages, it's pretty awesome. Because who gets to do that? Not a lot of people. The broad framework that I would like for my dissertation is not just like the lab bench work that will serve as like pieces of evidence to inform applications of science, but also the policy end of that. So obviously what I'm researching is a very tiny little nugget, but if we can start to or at least if I can add to the knowledge that we have on how to make ports or marinas less polluting for the animals that live in them, that'd be freaking cool. Especially if we can start to apply that to policy aspects. So how do you manage anti-fouling policy? So like the paint that covers boats, that is meant to kill things that try to live on it so that the boats can do what they're supposed to do, but that also can serve as a toxin for things that live in the water. So how do you use the evidence from understanding how it affects the health of the animals in order to inform how to better deal with and manage anti-fouling policy on boats?